Hello and welcome to another episode of Chemistry in Action. In our last lesson, we studied the biopolymers found in food, carbohydrates, lipids, and proteins. We learned that these are digested in our bodies and converted into other molecules that are used for growth and development and as fuel for our energy needs. Another type of biomolecules, the nucleic acids, are found in our food but are not required for our nutrition. However, they are found in all living things and are essential to all life. Today, we will find out why this is so. We will begin by examining the structure of the nucleic acids, DNA and RNA. We will see why they are called the molecules of heredity. We've got a lot in store for you today, so stay tuned while we pause for some reminders. When we see that children resemble their parents, we take it all in stride and say that it is a matter of heredity. If we had to pin this down to a single molecule, then we'd say DNA, or deoxyribonucleic acid. Genetic information is stored in DNA. This is relatively new knowledge. Nucleic acids had been known for a long time, but no one knew exactly what they were for. In 1944, Oswald Avery determined that DNA was a substance of heredity. This was an extremely important discovery, although not everyone may have realized it then. Why is DNA so important? Our DNA molecules contain all instructions for all the proteins, enzymes, and ultimately, all the carbohydrates, lipids, and other nucleic acids our bodies will make. So, as we've been doing in our study of biopolymers, let's first find out what nucleic acids are made of. Two types of nucleic acids are found in cells: ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Nucleic acids are polynucleotides. That is, they are polymers of nucleotides. A nucleotide has three distinct groups: a base, a sugar, and a phosphate. Let us consider these groups one at a time. First, the bases. The bases of DNA and RNA are nitrogen-containing aromatic compounds. Four types of bases are found in RNA: the purines, adenine, and guanine, which have a two-ring structure, and the pyrimidines, uracil and cytosine, which consist of a single ring. DNA contains the same bases, adenine, guanine, and cytosine. Instead of uracil, however, it has the base thymine. A closer look at thymine shows a methyl group at carbon 5. Otherwise, all other parts of the thymine molecule are identical with uracil. The sugar of RNA is ribose. Ribose is a 5-carbon sugar. Like glucose, it can be written as a cyclic compound. Note especially in this structure the hydroxyl groups at carbon 2 and carbon 3. DNA has a slightly different sugar, deoxyribose. It is almost identical with the ribose, except that it has no hydroxyl group at carbon 2. Because of this difference in sugars, DNA containing deoxyribose is called deoxyribonucleic acid. RNA containing ribose is called ribonucleic acid. Finally, the phosphate group. The phosphate group consists of one phosphorus atom surrounded by 4 oxygens. It was known that DNA was made up of bases, sugars, and phosphates. But how are these parts arranged in the polymeric structure of DNA? This remained a puzzle for many years. In 1953, James Watson and Francis Crick, using X-ray fiber diffraction data of Maurice Wilkins and Rosalind Franklin, proposed a structure for DNA. It was quickly accepted as a correct structure and is popularly known as a DNA double helix, or sometimes as the Watson and Crick double helix. The polymeric structure is built by alternating sugar and phosphate portions. The bases extend from the sugar molecule. A DNA molecule is formed by two strands of the sugar-phosphate backbone, like the sides of a ladder. The bases are on the inside and form the steps or rungs of the ladder. In this arrangement, each step of the ladder consists of two bases held together by hydrogen bonds. The DNA double helix is not an ordinary ladder, but resembles a twisted step ladder. Each sugar-phosphate chain forms a right-hand helix. The two chains are intertwined in a right-handed double helix. Scientists sometimes jokingly call one strand the Watson strand and the other the Crick strand. What kind of hydrogen bonding is found in DNA? Watson and Crick proposed that adenine forms hydrogen bonds only with thymine and not with guanine, cytosine, or adenine under normal conditions. Guanine pairs with cytosine. We say that adenine, thymine, and guanine, cytosine are complementary base pairs. Adenine, thymine, and guanine, cytosine base pairs are the most stable and preferred base pairing in DNA. In nature, DNA molecules are very long polymers. Human DNA has a molecular weight of 8 times 10 to the 10. The DNA of most bacteria is in the molecular weight range of 2 to 3 times 10 to the 9. How do such long molecules fit into tiny cells? Nature solves this problem by fitting this into a small space by twisting, packing, and compressing DNA around proteins. If we purify DNA in the lab, we break this protein-DNA association and obtain long fibrous DNA molecules like this. It is probably beyond the scope of high school chemistry to do a laboratory purification of DNA, although it can be done. Let's now go on to another characteristic of the double helix. Because of the complementary adenine-thymine and guanine-cytosine base pairing, one strand of DNA is a complement of the other. What exactly does this mean? It means this. If one strand contained all adenines, let's call it a poly-A strand. The other strand would contain all thymines, a poly-T strand. We say that the poly-A strand is complementary to the poly-T strand.